Hello, this is the summary of article which is created by Ferdaous et al. In 2020. The topic is about the CNN for cervical cancer, and the title is CNN-based approach for cervical cancer classification in whole slide histopathology image. This explanation is created by Fadhlan Afif and 16097017. So let's start with the introduction to cervical cancer. Well, cervical cancer arises from the cervix, so it is only occurred on women, and the pathogenesis involves HPV in 99.7% of cases, with 71% belonging to HPV genotypes, HPV 16 and HPV 18. And cervical cancer has two major types, SCC and AC. Routinely, only a microscope examination of biopsy samples conducted by certified board-certified pathologists can confirm the cancer diagnosis. And this diagnosis or interpretation may lead into some result. And the first is stage of the cancer, second the type of the cancer, and the third the choice of treatments that must be conducted. So the result of interpretation is really important. And on the other hand, it is also very challenging and error prone due to human eye brain system limitations. Even the picture resolution is really good, but we may not see something in really detail. And the second is fatigue. When we are doing same job over time, so we may feel fatigue and it affects the result. And the third is distraction factors during examination, or what happened in our brain, the pathologist's brain, may affect. And the fourth is non-informative regions. This is due to the extraction of the samples that will be explained later. And these days, WSI has been used, and this is the emergence of this use of deep convolutional neural networks in digital pathology. Now let's move forward and see about dataset acquisition. And the author retrieve HISTOPATHOLOGY WSI of ACC and AC, hematoxylin and eosin stain biopsy sample from TCGA. And hematoxylin and eosin is some kind of chemical substance that are used on biopsy sample, so the biopsy sample can be examined, can be interpreted. TCGA is a platform that stored a lot of data, a lot of biopsy samples, not only cervical cancer but also another 33 types of cancer. And the computational pathology requires high input image, so The author select only the formalin fixed paraffin FFPE because uh, it has good tissue morphology safeguarding, and thankfully the identification was carried out using the relevant metadata embedded in TCGA barcodes. So let's see how the data input looks like. This tree is the example of a tissue sample of the patient, and the artifacts, as can be seen here, is the result of alteration related to surgical removal, tissue processing, microtomy, digitization, and the example shown here is shows some issues that may lead into misapprehended unintentionally by pathologists and also the computer-based learning algorithm. In picture number A, the sample is marked by the marker or red marker, and this may lead into misinterpretations. And number B is that the tissue sample falls on top left area. The blue ink residues, air bubbles, and cutting artifacts. And number C is also there are a significant air bubble and heavily stained in some regions due to tissue faults. So these issues is come from the tissue processing or microtomy or surgical removal factors. So the author really control each slide manually as closely as possible to improve the classifications. About the stain normalizations, because of the tissue sample use the HNE as a nuclear dye and cytoplasm in histopathological diagnosis, so the color may become varies start from blue deep purple and pink red, and that may not affect the pathologist's interpretation in this case, but it have serious implication in computational medical analysis. So the author already discard low stain condition of WSI during QC phase as mentioned before, and the tiling method was centered while ignoring the remainder edge styles. So they use 1024 and 1024 pixels. Another step on processing the input is that there are irrelevant white space in non-tissue regions. So this process is often difficult because extracting the region of interest is usually manually operated. So the editor remove only full white tiles automatically by using the RGB values technique, but the rest was done customarily. In the end, 300 ACC and 300 AC tiles containing at least 90% of tissues is obtained for further training. So to sum up, In this experiment, the author used architecture that can be seen in table 1, and all the images were resized to 224 by 224 and split 70% for train, 10% for validation, 20% for evaluations. So you can see the summaries on table 2. And the important things to note is that first, they extract the stored values from model last convolutional layer with ImageNet pre-trained weights. And secondly, the deep learning extract features were used effectively in a subsequent fine-tuned VGG16 classifier after achieving 97% accuracy. In the fine-tuning process, they froze the first 4 convolutional blocks and redefined the last convolutional one, as well as the fully connected layer. And as main strategies, they use stochastic gradient descent as optimizer and binary cross entropy as loss function for 30 epochs in total. And the learning rate value is 0.0001 and batch size of 16. In the end, now we can see the performance on the test sets in table 3 as follows. And we may see that the accuracy, precision, recall, and F1 score shows good result. These experimental findings are remarkable and demonstrate the capability of deep learning pre-trained models toward promising computer-aided diagnosis in digital pathology. And this study is an initiation in light of fact that some cancer patterns cannot be cleaned by human examinations. And another thing to highlight is that how the dataset as input is processed. They control each input so it will not lead into misinterpretations. That is also interesting point here.